Right, good morning, good afternoon, and good evening depending on where you are in the world today. Welcome to the CPhI and P-MEC China E-Trade webinar, Bio Insights Cell and Gene Therapy. I'm your moderator today and we have our two speakers today, Peter Shapiro and Cameron Bardliving. So our first speaker today is Peter Shapiro, Senior Director of the Drugs Global Data and Platform Integration. Peter worked in small molecule drug development for Abbott Labs and on in vitro diagnostics at Embergen. He's also worked on the integration of Biopharm Insight, Lead Sheet, Pharma Sources, and IHS Market into Global Data's Pharma Intelligence Center. Peter is online, and over to him.
Hi, my name is Peter Shapiro, and I'm a Senior Director at Global Data Healthcare. And one of the groups I look after is the Pharma Source group, which talks about pipeline drugs and contract manufacturing. And I'm really glad to be part today of the virtual offerings for CPhI China. And today, my topic is Bio Insights, Cell and Gene Therapies. So I'd like to talk to you about that today. But before I get into that, I'd like to spend a little bit of time talking to you today about the Global Data Pharma Intelligence Center, which is our premium offering in the pharma competitive intelligence space and where a lot of the data that I'm going to go over today comes from, including the reports. And then I'd like to talk about trends in cell and gene therapy pipeline. I'd like to talk about regulatory designation, trends in cell and gene therapy. I'd like to talk about potential blockbusters in the cell and gene therapy pipeline. I'd like to talk about recent drug approvals in cell and gene therapy. I'd like to talk about forecasted sales changes in cell and gene therapy over the 5-year period. I'd like to talk about recent trends in cell and gene therapy contract manufacturing, as well as issues we're seeing in cell and gene therapy contract manufacturing. Then I'd like to go over the takeaways from this talk, and finally, some information if you'd like to contact us to get more information about cell and gene therapy or contract manufacturing or any of the other topics we're going to discuss today. So first, to go into the Pharma Intelligence Center. So this is our leading healthcare platform. And Global Data is the leading market intelligence provider to decode the future. The Pharma Intelligence Center, or Pharma IC, combines in-depth analysis, news, data, granular market intelligence to provide a complete view into the pharmaceutical industry. So examples of things that we've been tracking, some of which we're going to talk about today, include clinical trials, deals, pipeline analysis, contract manufacturing, response to COVID-19, as well as COVID-19's impact on pharma. We're also going to talk a little bit about sales, review designations, and approvals as well. So the Pharma Intelligence Center is a fully integrated online platform with reports, databases, and tools, strong analytic capabilities, automated alerts to embed into your workflow, and a single login. We have transparent methodologies, and all assumptions and data sources are captured in the reports and databases, full access to market forecast calculations, including epidemiology, segmentation, patent shares, and so forth, as well as specialty disease experts. So we have in-depth forecasts and analysis for over 20 highly prevalent, emerging, rare, and niche diseases, as well as commercial epidemiology, which is patient-based forecast models determine the market size for therapeutic indications, all delivered through one single platform. And today we're going to talk a lot about data that was in a report we published last year, the outlook for viral vector contract manufacturing, gene, cell, and COVID-19 vaccines. So first, again, to talk more about trends in cell and gene therapy to understand where we've gotten to today, we'd like to start by looking at the pipeline. So when we look at the global pipeline for drugs, of course, it's no surprise that we see the lead molecule type is small molecule, which makes sense, as that traditionally what drugs are and what most of the drugs you take every day are. But interestingly, when we look at the active pipeline-only drugs, we see that in the top 5 molecule types, we see gene-modified cell therapy, and gene therapy. And this is interesting. These are in the top 5, despite low approval numbers historically. Now, just to differentiate, the reason why we have these 3 types, not just gene and cell therapy, is because of the added demands of gene-modified cell therapy that we're going to talk about later in this presentation. So hold on if you have any questions on that. And also we see that industry and investors are really anticipating the promise of these new molecule types and various submolecule types that fall underneath these. So when we look at the active cell and gene pipeline-only therapies by phase, we also see something interesting. We see that while, of course, the entire pipeline is always weighted to early in phase, which makes sense because of the nature of the pipeline, that pipeline cell and gene therapies are weighted even earlier than other molecule types. And interestingly, gene-modified cell therapy is the largest component, as we saw before. And it's also weighted to the earliest phases of these 3 subtypes, which is interesting and a direct result of it being the newest of these subtypes. While cell therapy is the most advanced of these molecule types because it's the oldest when we look at it broken down by phase. And this is something that's really interesting. And sort of we see overall trends that were started by biologics and the nature of research really trending into these more complex and advanced biologics. So next, we want to look at cell and gene therapy, pipeline therapies by therapy area. Therapy area is our grouping of indication or disease. And almost half of cell and gene pipeline therapies are for oncology indications. And this makes sense because oncology is traditionally the most innovative therapy area. This is because of the need and of course the demand for oncology treatments. As many of these therapies are early stage, as we said before, the most popular oncology indication for these therapies is solid tumor, which of course is a very non-specific indication. As therapies go further through the pipeline, the clinical trial process forces them into having more specific indications. Interestingly, pancreatic and blood cancers are also popular indications for these cell and gene therapies. I want to also point out CNS is very popular, especially for cell therapy, and that's some of the historic understanding of neurology and stem cells is really hitting that up. As well, of course, we see genetic disorders, and of course, genetic disorders really has a really great map into gene therapy for obvious reasons. So when we look at the database and we look at drugs that have either been recently launched in 2022 or are predicted to be launched this year, we see a number of drugs. Currently, 8 future blockbuster drugs are expected to be launched in 2022 that we already see, 7 of which are in pre-registration, 1 of which has already been approved. this has been altered from the previous report in that one drug that was in our last report recently was withdrawn. But interestingly, if you look at the two in red, you see CAR T cell therapy by J&J, which was originally developed by Legend. And you see a gene therapy by BioMarin. So it's really interesting that this list includes both gene-modified CAR T cell therapy and a gene therapy. It tells you something about what we see even the near-term future predictions being. Then when we look a little bit further out into the pipeline, a good thing to look at are review designations. So these review designations are given by regulators to spur innovation. And the two regulatory bodies we're going to look at today are the US FDA and the European Union EMA. And there are many that we track. We're just going to talk about these two because just we have the largest real numbers of designations here. And we see many of these cell and gene therapies have received orphan designations, which makes sense, especially on the gene therapy and oncology fronts. And just want to drop a little thing because I know this is CPhI China, and cell and gene therapies have achieved 8 breakthrough and 1 priority designation in China. So there are other regulatory bodies. And wanted to point out the new Regenerative Medicine Advanced Therapy designation by the US FDA, which will benefit cell and gene therapies. And I just want to remind everyone that review designation is an indicator of regulatory interest, and global data has, among other people, have found this to be something that increases the likelihood of approval. And if you want to talk more about the likelihood of approval too, that's something I'd be happy to afterwards. So when we look at approvals for cell and gene therapies, we naturally look at the innovative drug approvals by the FDA because currently the US still leads the world in the approval of innovative therapies. And we can see there in orange the cell and gene therapies. And we can see the real, growth over time with the highest ever cell and gene therapy approval numbers in the previous year, 2021, with 4 approvals. That beats the 3 from 2017. And then, as we talked just in the last slide about the CAR T gene-modified cell therapy developed by Legend and Janssen. That's Carivicti, already approved this year. So really off to a great start. So when we want to look at other ways of sort of gazing in and decoding the future about cell and gene therapies, something we can look at are analyst consensus sales forecasts. So these are investment bank forecasts of drugs and therapies for the future. And when we compare the forecast numbers for current cell and gene therapies with those from 5 years in the future, we see the largest difference of any of the molecule types that we track. So here you can see the percentage sale changes over the next 5 years forecasted for the top 10 key molecule types. I just want to point out on top, we see gene therapies, which really are expected to grow the most, with over 12-fold or 1,200% growth. Next, gene-modified cell therapies like the CAR-T therapy we just spoke about, which expected to display the second greatest forecasted sales growth, with an increase slightly above 10-fold or 1,000%. Just wanted to point out on this, interestingly, we don't see mRNA vaccines appearing, even though they've been really quite successful during the pandemic. There really hasn't been a late stage pipeline yet that would really get forecasts going. So that's something to think about definitely for the future. That really wasn't something that we would anticipate, except for what's happened with COVID. So now that we've looked a little bit into the future and trends, we'd like to think about looking into the trends regarding contract manufacturing. After all, this is a CPhI meeting, even if it is virtual. So when we look at trends in contract manufacturing agreements over the last 5 years, by volume and by molecule type, we really see a growth in contract manufacturing service agreements for cell and gene therapies was hampered by COVID-19 for cell and gene therapy. However, we see a really dramatic growth for contract manufacturer agreements for vaccines, and this is a direct result of COVID. We have to understand that this is only a sampling of what's disclosed, which is not anyways a full view of the marketplace. And when we see the 2022 year-to-date, we really have to be aware that a lot of this information is sourced retrospectively from information after the drugs are further along. So it's very likely that number will come up significantly for both vaccines and cell and gene therapy. And this is something that we track in our deals database as part of our Pharma Source offerings. Also, when we're looking at contract manufacturing, and this is something we do on an annual basis, where we look at all of the new approvals and who are manufacturing and when that's disclosed, looking at the top API dedicated contract manufacturers for cell and gene therapies. And here we're only looking at those approved in the US, EU, and UK, which, while not the entire market, is sort of the more developed end of the market. We'd love to expand this in the future. We see really a limited number of API CDMOs manufacturing more than one approved drug, being led by Lonza, which has historic strength in cell and gene manufacturing, and in, of course, manufacturing of the machinery used for cell and gene manufacturing. There are really few companies with these sophisticated manufacturing capabilities, and there's a limited geographic distribution of these facilities, which is notable as these therapies often require cold chain transport. And I just want to point out in the other companies that are on this list, that Patheon, which is part of Thermo Fisher, here includes Brammer Bio, which was acquired by Thermo and made into Patheon Viral Vector Services. And we're going to talk about viral vectors in a little bit more depth later. So transitioning to that, we want to talk about issues in cell and gene therapy contract manufacturing, and talk a little bit about viral vectors. And first we have to understand, make sure everyone has the same understanding of viral vectors. So we want to talk about gene therapy and gene-modified cell therapies first, because that's, in our topic, of course. So these, of course, are exciting and complex new treatments. We often hear them referred to as regenerative medicine or ATMPs in Europe. They involve inserting foreign genetic material into a cell to prevent or fight disease. They really have high price tags because of this. We have a small number of approvals, as we've discussed previously, and many, many more hopefully to come, and potential for a great many indications. And including in this, we see gene therapies, such as the world's most expensive drug for a rare orphan indication. And also, we see these more complex gene-modified cell therapy, CAR-Ts, like we just discussed before. And then for recombinant vector vaccines, this is like really the newest of the new, where we see the first approval in 2020. And these deliver the DNA into the cells of the body, and then the DNA tells the body to make antigens, much like mRNA vaccines deliver mRNA into the body. And this is unlike traditional vaccines that contain antigens already. And there's really a lot of potential for their use in preventing infectious disease or treatment of cancers. And here, first, but definitely not least, we see J&J's set of Ebola vaccines, which were the first approvals. We see AstraZeneca's COVID-19 vaccine. We see J&J's COVID-19 vaccine, and Russia's Sputnik vaccine, among examples of recent recombinant vector vaccines that have either gotten approval or emergency use authorization. And both of these sets of drugs use viral vectors. So something that there was already a shortage of, which we're going to explain, now becomes even more of a shortage. So when we look at the geographic spread of viral vector manufacturing facilities worldwide, we really see that these facilities are quite concentrated. Now, I will put a caveat that this is from our report that was a year ago, and we have seen some spread since then. So a majority of the sites are still in the US, but we have seen CMOs and CDMOs start to build capacity for viral vectors. But really, compared to monoclonal antibodies or more traditional biologics, there really is relatively little capacity worldwide. We have less than 100 contract viral vector facilities worldwide, and we're going to talk about how we've seen the spread. We see the UK in the second spot, and this is thanks really to active government funding, and very few sites in the Asia Pacific region, which is something we're going to talk about how it's been changing. And no contract sites in South America or Africa. So next, we're going to look into the approved products that use viral vectors. And so back to our topic of cell and gene therapies, we see 19 marketed gene and gene-modified cell therapies that use viral vectors. And even before the pandemic, there was a shortage of viral vectors for these regenerative products. But now we have the addition of 9 recombinant vector vaccines for COVID-19 and Ebola. And demand for COVID-19 vaccines really dwarfs all the other demands and is pushing up viral vector demand. So here, when we look at a global data COVID-19 dashboard that we have in the Pharma IC. That's our premier intelligence offering in the healthcare space. We see an idea of really quite dramatic number of doses that are required. And then if we think about the number of genomes per dose, really how large this manufacturing feat is going to be. So our model shows that the current bioreactor capacity worldwide is really a minute fraction of the capacity that we'll predict will be needed just for the viral vector products that we see in the current pipeline. This includes both gene therapies and gene-modified cell therapies, as well as recombinant vector vaccines. So really, we're going to see the need for significant growth of manufacturing. And we've already started to see this. So I've read on this slide a couple times now because we've continued to see growth. I read you this to sort of feature the APAC region a little bit more because, of course, this is CPhI China virtually, and really want to address that. So we see GeneScript, which is adding a very large plasmid viral vector manufacturing facility to an existing site in China. We see Fujifilm, which has been very busy in the US, will build a campus in the UK for gene therapy and mammalian cell culture. We see UCB, a real European powerhouse, building a gene therapy facility in Belgium. And the Center for Breakthrough Medicine has received equity financing from SK, existing CDMO, for preclinical through commercial manufacturing of cell and gene therapy. Last but not least, Samsung Biologics is investing in a super plant in South Korea to break into the vaccine and cell and gene therapy markets. And really, this is just a small portion of what we've seen in the last couple of years. As I said, we've redone this slide several times now for several different events. As well, we're going to need to see processing breakthroughs in cell and gene manufacturing. So upstream improvements in viral vector manufacturing could really increase production quite a bit. And current generation of cell lines is really inefficient and costly. Improving productivity from better cell lines and processes. We also think about how to improve scale. And this is especially from switching from adherent cell lines to suspension in culture, which really would help the volumes. As well, we want to think about downstream improvements after the vector is grown in recovery, which would really increase production fivefold. Viral recovery is really a challenge, and improvements in purity must be balanced in mitigating losses to keep as much of the starting material as possible. As well, we're seeing the development of next generation chromatography materials, to separate. And as well, we're seeing sort of regulatory changes, so future acceptance of common platform off-the-shelf vectors. This would avoid study duplication, which would really reduce demand. And really is what we see for other types of drugs, such as small molecules and more traditional biologic like monoclonal antibody manufacturing with interchangeable platforms. And will really reduce the time for tech transfer, making it significantly easier. So Peter Marks, the head of CBER, the FDA, has talked about the use of the same vectors that can be manufactured and characterized by well-defined processes that have been used previously. And that this would be a vector would be like the razor, and the insert would be like the razor blade, in the case that you can then define the razor as accepted and then just test the different blades. One could reuse information on the vector backbone to expedite the development process of multiple gene therapies. Of course, there'll need to be some caveats to this. So then I'd like to go into the takeaways. So first, I'd like to go into our key findings that we've talked about. So I'd like to talk about industry interest in cell and gene therapy. So we see approved drugs dwarfed by this really massive pipeline of cell and gene therapy. Concentration of the cell and gene pipeline in these early pipeline stages, and that this is dominated by oncology, which is traditionally the home of more innovative drugs. And we see interest by investors and regulators for cell and gene therapy. So we see future potential blockbusters in the late stage pipeline and in recent approvals. We see high sales forecasts really changing dramatically in 5 years in the future from what we currently see for cell and gene therapy. And we see that new review designations have been created. Then when we look at cell and gene manufacturing demand, we see there's a shortage of viral vector manufacturing. There are inefficiencies in manufacturing that are being worked on, and that regulators themselves realize that they've yet to catch up with the new realities of cell and gene. So what to watch for? More pipeline cell and gene therapies, more review designations granted to these cell and gene therapies, and a greater variety of therapy areas as this technology sort of trickles through the system. This will lead to more approval of cell and gene therapies, and an increase in revenue from cell and gene therapies is also predicted and will, of course, be needed to drive this because of the great expense of development and manufacturing for these drugs. And last but not least, more manufacturing facilities with more capacity, more efficient manufacturing to be developed, and regulatory changes and convergence from multiple markets. As more products are expected to be launched in the next few years, there'll need to be more faculties geared towards manufacturing marketed viral vector therapies with the right quality systems, as we got as a quote from KOL. So last, just to remind you a little bit about global data. We employ only the best academic and critical thinkers from the healthcare industry to ensure you receive unrivaled analysis, support, and solutions. Our expert healthcare team consists of 400 researchers, analysts, senior analysts, project managers with hundreds of years combined experience. Global data provides you with the most extensive range of pharmaceutical contract manufacturing databases, analysis, insight available from their origin as a Pharma Sources, which company we acquired now 5 years ago. And we've drawn on a number of reports and databases that are on our platform for this talk. Point out a couple of them that we recently have published, new drug approvals and their contract manufacture, and future of pharma looking ahead to 2022, as well as some more historic Pharma Sources reports from last year, including viral vectors that we talked about at the end, and COVID-19 contract manufacturing, as well as our monthly biopharma outsourcing offering and our quarterly emerging markets outsourcing report. So please feel free for any questions or further inquiries, please contact us at clientsupport.globaldata@globaldata.com. Thank you very much. I hope you enjoyed this talk.
Thank you, Peter, to talk with us for your insight, and we'll move on to the second part now. Let's introduce Cameron Bardliving, who's the Director of Operations from Jefferson Institute for Bioprocessing at Thomas Jefferson University. So Cameron, over to you and good luck.
Hello, my name is Dr. Cameron Bardliving, and I'm Director of Operations at the Jefferson Institute for Bioprocessing. And thank you for having me today at your conference. Before I begin, I just wanted to give a high-level overview of our capabilities here at the Jefferson Institute for Bioprocessing and really just describe to the audience here about what we do here at JIB. We are a 25,000 square foot GMP simulated flex factory where we have three main functions and goals. Our first goal is to educate the next generation of talent for biopharmaceutical manufacturing and process development in our facility, whereas we have master's degrees, PhDs, postdocs, certificates, all designed and focused on industry, and particularly the biopharmaceutical industry. We also do many many days of industry training for some of our large pharma companies and other small companies. Those trainings can be introductory for operators and technicians. It can be more advanced for some of the process scientists. But again, all of it's focused on biopharmaceutical manufacturing and development. As you can see here, our largest reactors are 200 liter scale, which you'll see in a moment, or you probably just saw. Again, this entire facility is all single-use, flexible, ready-to-use technologies, really the next generation of manufacturing for biopharmaceuticals. And lastly, we have capabilities to do development work for small companies who are looking to evaluate preclinical biophysical characterization, tox material for new molecules. And that could be in monoclonal antibody, replacement protein, viral vectors, cell and gene therapy, advanced vaccines, stem cell derived therapeutics. You name it, we could probably handle it in our facility. In addition to what you see here, we are also expanding our facility to include more what we call BSL-2 laboratories that do more cell and gene therapy work. In addition, more analytical capabilities, which will be housed now in a new separate facility. So again, as you see here in the slide, lots of investment from Thomas Jefferson University, and we are really moving forward in this next year to grow our space and grow our team. So exciting times here at JIB. So the first part of my talk really is want to lay the groundwork for what's the problem and why are we all here and what is the problem here. So at the turn of the century, there was lots of promise with the human genome sequenced, and really the community was really thought we can, and we still are, and we'll talk about that more, is now we're moving away from the old style of treating diseases now to looking at this next generation of therapies and addressing potentially curative solutions for some of these catastrophic diseases. And so for this, as this child is an example, whether this child is in Philadelphia, Peterborough, Punjab, or Panjim, the future is looking bright for this potentially for this individual, for these types of these next generation of people on this planet, where we can now move from the old way of treating therapies, for example, in this case is cancer, moving away from radiation, surgery, and chemotherapy, which is the three standard ways of treating cancer, to potentially some new fourth way of treating these disease states that will allow a patient to have a better quality of life, more effective in treating these disease states, and obviously, hopefully, lead to a better prognosis. And if you look at the major cause of disease in the world, it's not just cancer. We have a host of cardiovascular diseases, cancers, obviously, other forms of diseases and death, including neurodegenerative diseases, Alzheimer's, for example. In the last couple of years, we've all experienced the infectious disease, the parasitic diseases. So in this case, there's lots of areas where we need to do work. And in this case, what we're going to focus the talk on today is the area of immunotherapies. And in this category, I'm going to include protein replacements, cell therapies, and gene therapies. So this slide really tells us how did we get here. This is not an overnight activity. In fact, this goes all the way back to the 1950s. We could consider the discovery of the structure of DNA one of the first step towards where we are today. And I highlighted here two what we call annus mirabilis, these miracle years in our space in biotech. One being the recombinant DNA technology created in the early 1970s that led to the over the next decade after that to finally the production and license of the first synthetic insulin Previously, insulin was harvested from pig and pancreas sources, pig and cow pancreas sources, and by 1982 we were able to develop a fully recombinant recombinant DNA-based biopharmaceutical for human insulin for human use, and that's obviously still on the market today by Eli Lilly. And so over the next 30 years, we've grown from just doing insulin and smaller proteins to now we have a host of monoclonal antibodies and other more complex proteins on the market. In the early 2010s, we have the first gene therapy approved for use by the FDA. And then in 2015, which is we consider another miracle year, this is the CRISPR-Cas9 technology. It's the breakthrough year for gene editing technology. It's really transformed what we could do, and now we were able to go into the disease state and really alter the expression of the expression patterns of these proteins. And so now we're just starting to see the first CRISPR engineered gene therapy, cell therapy correction trial is underway. So this is a really exciting time to be in this space. We have another example here is the advanced cell therapies to produce insulin. You can see what we've gone from in the early part of the century, where we were harvesting it from animal sources, to now we're able to produce human cells that go back into the patient and potentially lead to a cure for the disease. So again, lots of exciting things going on. To get to this fourth way, this immunotherapy approach, this cell therapy and gene therapy approach, you really need 7 elements to be successful. It's not just about, obviously we see here in this diagram I have on the slide, understanding to produce a high quality medication, we have to understand the mechanism of action, show me in case safety and efficacy of the molecule, understanding of the process, what are the quality attributes of our molecule and our therapeutic. And really to get those out to that state, you need several things. You need good clinical operations, good translational research. You need advanced technologies and facilities to produce that medication. You need obviously investment from large companies potentially. And then obviously define efficient and effective and scalable processes, right? And so altogether, this allows us to assure of supply of our high quality medicine. So it's not easy. This is not an overnight activity, and it requires. In addition, I forgot to mention skilled workforce. It shouldn't be an afterthought, but skilled workforce is obviously needed to produce these medications. And so altogether, this allows us, again, like I said earlier, to supply high quality medication to our patients. So this is not something that we haven't done before. We've been very successful with monoclonal antibody therapeutics. And if you look at the growth of this space over the last 30 years, it's been incredible. This is life expectancy gains estimated to be worth over $3 trillion a year. We were able to treat a variety of cancers now. Every day there's something coming out of the market. So this is a very positive sign for patients and obviously for the growth of this industry. We've also gotten very good at manufacturing monoclonal antibodies. We have a platform technology now. So if you look at the cost of goods for a monoclonal antibody and traditional protein replacement therapies, initially in the mid-1980s and the early 90s, we were at $10,000 per gram, very low titers, and we've improved that tremendously over the last few decades to now where we're talking about $100 per gram, maybe even less, and upwards of 10 grams per liter titer for some of these proteins. So, and this has all been accomplished through improved technologies, more efficient processing, platforming, like I mentioned, because we have similar types of molecules going through similar processes. And obviously, hopefully that's been translated to the patient. And again, this is a remarkable time to be in this space. And again, here, this is just a plot here of the market value from 1975 to a couple of years ago for monoclonal antibodies therapeutics. And you see here some of the key therapeutics that are highlighted. This is obviously not an exhaustive list, but this shows you some of the key players here, and some of the direction of this arrow looks very nice for our finance colleagues. So here, this is going upwards of potentially $300 billion a year, maybe more by 2025. So again, a good time to be in monoclonal antibodies. Again, I'll briefly go over this slide here. Again, lots of investment, and this is just for the US. So millions of jobs, upwards of potentially 5 million people working in this space, great salaries, a huge contributor to the US economy, lots of molecules in development. A lot of them are first in class, and primarily focused on cancers in this case and heart disease. So again, growing space, lots of opportunities. And again, for our students that come through our programs, we tell them this is that this is a good time to be in the large molecule space and biopharmaceuticals in general. Now, as we move to cell and gene therapy, we have to address challenges that we had to address for large molecules and specifically monoclonal antibodies. So as we move from small molecules to insulin and human growth hormone to monoclonal antibodies, the complexity of that molecule, the complexity of that structure-function relationship escalated as we moved up the ladder. And now that we have cell therapies and these gene therapies, it's really going to the next level. We can even consider it exponential in this case. And so what we really need to do as the structure and function relationship becomes more complicated to resolve fully, the regulatory agencies are expecting us to have a very tightly controlled manufacturing process and highly resolved analytical methods to understand the relationship between our process and the quality attributes of a molecule, how the role that our processing will play on that quality, because if we can't fully characterize, we need to make sure that we have a tightly controlled process, right? So it's about reducing, understanding our residual risk, and then also reducing it through a robust control strategy. But all said together, I outlined the challenge here, but really I would like to say that we've made tremendous progress over the last century, going from harvesting insulin from animal sources to, in this case, I show an example of a fusion protein, which is a once a week insulin regulator from I guess Eli Lilly in this case, to now, as I mentioned before, this islet-based cell therapy, where this is almost once in a lifetime treatments. So we go from multiple times a day to once a week or a month, all the way to once in a lifetime treatment. But what we need to consider now, as we move into this next generation of therapeutics, is think about the cost of therapy, think about the speed of getting these medications to the patients, think about the product quality, and think about the patient quality of life. All of those things need to be thought about and considered as we develop this next generation of therapeutics, as we did for the previous generation. Right. So again, the list is very, very long here. This is just another example. In this case, this is Parkinson's disease. Can we develop therapeutics where we can have potentially a source of cells, which we can, in this case, it could be a stem cell, could be another source. We could differentiate these to in this case, a particular neuronal cells product, but it could be anything, right? It doesn't have to be just this example. And there's many of our colleagues that are working in this space. The science has been worked out, the discovery has been worked out, but really the challenge here is, how do you manufacture these at scale? How do you produce them robustly? How do you maintain product quality? How do you address the cost of the therapy? How do you address the speed of getting this therapy to the patient, right? All these need to be are considered and still remain big challenges for these next generation of advanced cell therapies. Again, more going over the fourth way. This video I have up here is a cancer cell being killed by T cells. Again, this is amazing discovery, amazing science. And again, here on the slide, we have some of the 4 categories. We've shown that we can do gene replacement therapies. We've demonstrated that we can do gene silencing. We've demonstrated gene editing and gene addition. Right. If my next slide here is going over some of the real examples, right? This is not something that just is theoretical. This is not something that's still in the academic lab. These are on the market. These are close to being or in clinical. Again, in the area of gene replacement, siRNA for gene silencing. The CAR T is really gene addition, with several approved products in the CAR T space. And then, as I mentioned earlier, CRISPR-Cas9 for gene editing is just getting into clinical. So lots of breakthrough therapeutic platforms. And again, like my take-home message here is, how are we manufacturing these? How are we providing consistency, speed, and addressing cost issues? Again, this slide here is just showing this space is very, very active, not only from the smaller companies, but a lot of the major companies here, the names, are all known. But this is just showing you, I like this, we call it the galaxy here, that, that this is not a dream. This is not something that is potentially in the future. This is the now. And some of the these major players are in this space now, developing some of these next generation therapeutics. Right. So strong foundational outcomes. This is again this cell therapy case. This is just a case study here with Kymriah. This is a example of somewhere where we need, there's potentially more work to be done. Right. This is wonderful treatment, but in this case, some of the challenges here were product quality, speed to patient, and patient quality of life. So, for example, in this case, and this is not to pick on Kymriah, but this is looking at the time from withdrawal of the T cells to reinfusion was about 52 days. For a competitor product, Yescarta, you see here that that same process took 29 days, right? In that 52 days, the patient could have a different disease state, progression of disease, impacting some of the outcomes of the patient for the product. And so, in this case, this demonstrates that not that science is fine. The science is well understood and established. In this case, the processing of that material has some challenges. And again, these companies are addressing these. And I've seen reports of even faster CAR T manufacturing processes. So again, the community is addressing this in an effective way, and I'm excited to see that this is starting to be addressed now that these technologies are becoming more mature. And it's all been established by improved technologies from our vendors, better understanding of the impact of our process on our product quality, and really a drive to get these medications to our patients in a more efficient manner. Another thing here is that another case study here that we have is gene therapy. So we've worked out the foundational scientists, very good clinical outcomes, but in this case, the cost is an issue. So a lot of these gene therapy products have upwards of hundreds of thousands, even close to $1 million, $2 million for a single injection. And so the question we have to ask ourselves is, as these prices are there's no secret here. These are significant issues for some payers and for patients to gain access to these therapies. And so some of the cases here, the manufacturing is cost of goods is very high, and it's also difficult to treat very large populations because of the cost of goods and the inefficiency of the manufacturing processes. So in this case, this is one area where we can make some improvements. Regardless of how we price these drugs, we can make some improvements in cost of goods here. And again, there's active research to bring the cost of goods down for these gene therapy products. So I just want to leave the group here with a excellent quote, I think, from the former former commissioner of the FDA, Scott Gottlieb. And really, you look at in his 2018 report, mentioned that if you look at the traditional drug review, about 20% of the emphasis is on CMC and 80% of it is on the clinical. But when you look at cell and gene therapy, it's really flipped. 80% of it is CMC, chemistry, manufacturing, control, and about 20% of it is on the clinical. And what that really tells us is that there still needs to be more work done to identify our understand manufacturing processes, address challenges in our manufacturing processes, and how that links to quality of our molecule. And again, so if you look at the applications for these therapeutic drugs and small molecules, you see a lot of the emphasis is on the CMC side. So long story short, again, this is a active area, a exciting area, exciting time. And I think the take-home message here is that we've gotten past the clinical, we've gotten past the discovery. This next phase, as we mature as an industry, is really to address some of the manufacturing challenges. And I'm glad to say that here at JIB, we are doing that. And obviously, our colleagues and friends in the field are doing the same. And I think the future looks very bright for these therapeutics. And I could definitely say that over the next 20 or 30 years, we'll be able to address these challenges. So with that, I'll say thank you for having me. And I hope you gained a lot of insights today. And please feel free to reach out to us in the future if you have any additional questions. Thank you.
Thank you, Cameron, very much for taking your time, and great to get your insight and experience. Everybody, thanks very much for joining and listening. Wish you a good rest of your day. See you next time.